let us assume this is a ribosome particle. This is a ribosome particle. This is the larger subunit. This is the smaller subunit. When the mRNA, let us assume this is the mRNA chain. The mRNA now aligns at the junction of the larger subunit and the smaller subunit. In the ribosome, there are two sites. One is known as the A site, the other one is known as the P site. A site, amino acyl site, and P site peptidyl site. Suppose this is the first codon in the mRNA. This is the caput region. This is the first codon. The first codon invariably has to be AUG. So this is the initiator codon AUG. This initiator codon codes for methionine. Therefore, only the amino acid methionine has to be brought here by a tRNA molecule, and that tRNA molecule will have an anticodon UAC. This is the 5' end, 3' end of the mRNA. This is the ribosome. This is the initiator codon. So now a tRNA molecule with an anticodon UAC, with an anticodon UAC carrying the amino acid methionine. So charged tRNA with UAC as the anticodon carrying the amino acid at the amino acid binding site methionine reaches the ribosome at the A site. UAC and AUG bind with each other. So the first amino acid has been brought to the site of protein synthesis. This is called chain initiation of the formation of a polypeptide chain. This requires three factors called initiation factors. three initiation factors, IF1, IF2 and IF3 are known to be involved in the process of initiating the formation of a polypeptide chain. So first amino acid has been brought to the site of protein synthesis. Now the ribosome slides towards the 3' end. As the ribosome slides, the second codon now comes in contact with this is the second codon. Second codon now comes in contact with the ribosome. Suppose the second codon is UUU, phenylalanine must be brought there. Phenylalanine can be brought there by a tRNA molecule having an anticodon A. So a second tRNA molecule comes here carrying phenylalanine. When the second amino acid comes to the site of ribosomes, a peptide bond is formed between the first amino acid and the second amino acid. A dipeptide is formed. As soon as the dipeptide is formed, this tRNA molecule, the first tRNA molecule, detaches and goes back to the cytoplasm. Again, it may come back carrying methionine if anywhere else the codon for methionine is present. Then ribosome slides, third codon comes in contact with the ribosome, third amino acid is brought by a tRNA molecule, third amino acid is attached to the second amino acid, a tripeptide is formed, the second tRNA molecule detaches. So like this, amino acids are added one by one. So after some time, you have a situation. This is called chain elongation. Chain initiation followed by chain elongation. mRNA is here. First codon, second codon, third codon, fourth codon, fifth codon, like this. At the first codon, methionine was brought. Methionine was brought by a tRNA molecule. At the second codon, let us say, phenylalanine was brought by another tRNA molecule. Methionine and phenylalanine are joined with one another. Then another amino acid, third amino acid, tripeptide, fourth amino acid, tetrapeptide. Now the ribosome is here. Now the ribosome is here. There is another tRNA. With a fifth amino acid that will bind here. Fifth amino acid, sixth, seventh, eighth, like this. So chain is elongating. This step is called chain elongation. This also requires certain specific factors which are known as elongation factors. Three such elongation factors have been identified: elongation factor 1, elongation factor 2 and elongation factor 3. the process continues like this. Finally, you have a stage. This is mRNA. These are the nitrogen bases that represent the triplet codons. Let us say at the other end, at the 3 end of this mRNA chain, at the 3 end of this mRNA chain, there is a combination of UAA followed by a poly A tail. UAA is the last codon present here. Remember, UAA is one of the 3 codons that does not specify any amino acid. So here, for each of these codons, an amino acid has been brought. All these amino acids have been attached to one another by a peptide bond, resulting in the formation of a polypeptide. So this is the polypeptide consisting of amino acids. Finally, here for this nonsense codon, for this terminator codon, there are no tRNA molecules. There are no anticodons, which means no tRNA molecule can come and attach here. No more amino acid can be attached here. So when no more tRNA molecule can come here, it simply means that the formation of this polypeptide chain is completed, and this is called chain termination. Chain termination. Chain termination also requires 3 different factors: termination factor 1, termination factor 2, and termination factor 3. So chain initiation followed by chain elongation followed by chain termination. The polypeptide chain, this is the polypeptide chain. Formation of polypeptide chain is complete now. This polypeptide chain is released. This mRNA now the ribosome is here. Ribosome is here, but no more tRNA can come, so no more amino acids can be attached. Now the mRNA comes out of the ribosome. The 2 subunits of ribosome again dissociate. So here they had associated with each other, here they dissociate. Polypeptide chain is released, mRNA is released, ribosome dissociates. Now this mRNA may again become associated with another ribosome, and may repeat translation. Again chain initiation, chain elongation, chain termination. Second molecule of the same protein. Third ribosome again translation, chain initiation, chain elongation, chain termination, third molecule of the same protein. So normally only one transcription, only one mRNA is formed, and that mRNA can enter into any number of translations, each with a ribosome, resulting in the formation of many molecules of the same protein or same polypeptide. There is another possibility. We have seen that in the cell, some branches of endoplasmic reticulum contain on their branches a series of ribosomes. A series of ribosomes. This situation is called polyribosome. Such a branch of endoplasmic reticulum is called rough endoplasmic reticulum. Rough endoplasmic reticulum. It is also possible that an mRNA chain may become associated with the first ribosome in a polysome, or a polyribosome may begin translation here, chain initiation followed by chain elongation, then the same mRNA will be coming in contact with the second ribosome, chain initiation here, chain elongation here, third ribosome, chain initiation, chain elongation, chain further elongation here. Like that it may continue, so that the same mRNA can become associated with a series of ribosomes, continuously translating several molecules of the same protein together. With individual ribosomes, translation may happen, or with a group of ribosomes, translation may happen. These are with ribosomes found freely in the cytoplasm. These are with ribosomes found associated with branches of endoplasmic reticulum. The proteins synthesized in individual ribosomes are released into the cytoplasm. They function as cellular enzymes after suitable modification, whereas the proteins synthesized in polyribosome are transported through the branches of endoplasmic reticulum to serve various other functions in different parts of the cell. So this is the mechanism involved in translation.